So we're gonna start with Ludovic. You here?
Yeah, hi. Yeah, I'm here. I'm the warm-up.
Okay, you are the warm-up. Okay. All right, warm up the stage.
Can you see my screen?
Yeah.
All right, awesome. All right, so my name is Ludovic Mouttet. I'm a master's student in my first year, and I'm part of Dr. Renzo Ciceri's lab. And I will present you the effects of doxorubicin on patient-induced pluripotent stem cell-derived cardiomyocytes. So just a quick agenda of what I will talk about today. I'll give a short introduction on the topic. Then I'll go over the methods and some of the preliminary results we have, and then talk a bit about the future directions we want to take. So first things first, doxorubicin is a chemotherapeutic drug, part of the anthracycline family, and it is used to treat hematological and solid tumors in adult and pediatric patients. However, dox is well known to promote doxorubicin-induced cardiotoxicity, which poses the most significant risk for cancer survivors to develop irreversible cardiac dysfunction and also heart failure after treatment. And all of these are unfortunately incurable. So there are multiple mechanisms that have been postulated for how doxorubicin works, but unfortunately, there are also still many unknowns about how it enters the heart. But some of the mechanisms are epigenetic alteration, calcium dysregulation, radical formation, regulated cell death pathway dysregulation as well, just to mention a few. So this is part of the Heart in a Dish project, where we collect blood samples from patients, and then we try derive from those blood samples induced pluripotent stem cells, and then have obtained cardiomyocytes. So far we have 390 blood samples that have been collected and stored. 210 of them are unique, and 180 have been withdrawals, and we have tried to have a 1-to-10 ratio of healthy donors to diseased patients. Of these 210 samples, 31 induced pluripotent stem cell patient lines have been generated. 6 of these are control patients, and 25 are diseased patient lines. And from these 25, 10 of them are doxorubicin induced cardiomyopathy patient lines. So our method is that we're looking at healthy controls, dilated cardiomyopathy patients not due to doxorubicin, and dilated cardiomyopathy patients due to doxorubicin. We then draw blood, and from that blood, we try and isolate peripheral blood mononuclear cells, PBMCs. We then transfect pluripotency factors using episomal vectors or Sendai virus, so that we can obtain induced pluripotent stem cells, iPSCs. We then culture these iPSCs, we expand them, we maintain and purify. Here on the top, you can see iPSC, like culture. And then these iPSCs, we try and differentiate them into cardiomyocytes using the GWE differentiation protocol with RPMI and B27 media. So once we have these iPSC-derived cardiomyocytes, we try and replate them, and to then do various assays. And what I'm concentrating on is incubating these plates with doxorubicin for 24 hours, and then do some assays to see the effects it has on the cells. And just, sorry, I skipped, but I have a quick video of how our cardiomyocytes look like under a light microscope. Here, they're just beating. So what's very important is that we are able to ensure purity. And so we do that by doing immunofluorescence staining. First, we do immunofluorescence staining for our induced pluripotent stem cells using pluripotency markers to make sure we have indeed generated iPSCs. Some of these iPSC markers are OCT4, SSEA4, NANOG, and TRA160. We also do immunofluorescence staining with cardiac markers to make sure we have obtained cardiomyocytes. And so the cardiac markers we try and look for GATA4, SERCA2A, troponin T, and connexin 43. So we're doing a chemotoxicity study, and to date, we have done 6 doxorubicin iPSC cardiomyocyte lines. We also have some healthy controls and dilated cardiomyopathy patients not due to dox. And we want to have these lines, we try and have these patient lines under normal conditions, and also we want to apply doxorubicin and see what happens to our cardiomyocytes using various assays. So some of the assays we use are a viability assay to ensure that the cells are alive and also to check the degree of cell death after injury. We also do a hypertrophy assay to check the effect on cell morphology. And finally, we do a calcium flux assay where we verify the contractility and the calcium handling dysregulation in response to doxorubicin injury. As I showed on one of the mechanisms, dox seems to dysregulate calcium, which would lead to cardiac toxicity. Now, also, we want to study some apoptotic and other cell death markers, as there are many pathways that dox actually impacts from the regulated cell death pathway. So what we aim to achieve is first to determine the relevant doxorubicin concentration. We have some preliminary data that I will show in the next couple of slides. We also want to establish an iPSC cardiomyocyte expansion protocol. Actually, a group from Stanford, led by Maths and Al, have published a protocol for this in 2021. And this is very novel because cardiomyocytes are defined by the fact that they cannot differentiate and expand. So this would make, if we were able to expand our cardiomyocytes, it would make studying this very easier because we could have a lot more cells. And finally, we would want to understand whether there are patient-dependent variability when treated with doxorubicin that can predict clinical outcomes. So now I will show some of the preliminary data we have. So first, we did a viability assay using crystal violet, and we saw that increased doxorubicin concentration reduces cell viability. As we can see, there's less crystal violet uptake as we increase doxorubicin concentration, which means there are less cells viable. Furthermore, we also did a hypertrophy assay where we found a trend towards a larger cell area and higher concentrations of dox. However, it is not significant yet, as this may be attributed to the lower number of cells we've measured, as it is only preliminary data so far. But initially, we did expect larger cell area because in response to injury, cells usually grow. And our final assay that we've done so far is calcium flux, where we see that doxorubicin reduces contractility. And the calcium flux, as here we can see, the control has a higher intensity of a calcium channel than our various concentrations for the experiments. So just quick videos here. You can see the normal concentration beating, the normal concentration intensity, calcium flux. And here are different concentrations of doxorubicin when they're applied. And we can see that it is less than the normal condition. So with this data, we will use between 0.05 micromolar of dox and 0.1 micromolar of dox, because it gives us the best cell viability, as well as the difference in, for example, the calcium flux and also hypertrophy. So this would be good to then continue doing our experiments, but we're not, and we probably will use between that range and concentration of dox. So some limitations we have is that, while we've had a healthy control group as well as dilated cardiomyopathy group whose disease was not caused by dox, we unfortunately don't have an additional group of patients who were treated with dox, but did not display any acute or chronic symptoms. Another limitation, it is that patient-specific iPSC generation is very complicated, and it also takes a long time. It takes us about 3 months, so it's hard to have enough power and cells to determine if some changes in our experiments are due to a change in protocol or due to a change in the cells. And also we have patient inter and intra variability. Finally, some future directions is that we want to increase patient sample size. We also want to do more assays such as MitoTracker to look at our mitochondria, what happens to it under doxorubicin conditions, as mitochondria is very much involved in it. And also we might want to use siRNA to do some experiments and interrupt some of the pathways. Also, we would like to explore various mechanisms responsible for doxorubicin-induced cardiotoxicity, such as, just to name a few, apoptosis, ferroptosis, pyrrhoptosis, and necroptosis. So this is all possible thanks to our team, who I would like to thank. And also thank our collaborators, Dr. Nadia Gianetti's lab and Dr. Terry Herbert's lab. These are some of my references, and I'm open to any questions you have. Thank you very much.
Thank you, Ludo, for your presentation. Now I invite the audience to ask Ludo some questions. Please raise your hands or ask your questions in the chat box and I will read them out. Thank you.
So Ludo, can you comment on the mechanisms that lead to the reduction in contractility and calcium flux, those wonderful results you have there? What is there a mechanism behind it?
We believe that there's less calcium uptake due to changes in, I'll just go back to just a quick image. Due to our smooth endoplasmic reticulum damage, so there's a bit of problems with the calcium exchanging genes. So that would lead to like the calcium channels have some are being interrupted due to the DOX, and then that causes the calcium dysregulation.
Okay, thank you.
Okay, Mashid asks, well, thanks for the presentation. How do you see the implications of these findings in clinical settings?
Thank you, first of all. And that's a good question. Well, in the clinical settings, it's a bit complicated for the moment. We're just generating the cardiomyocytes and we want to keep on expanding our patient size, but we hope to try and understand what may be some of the genetics of patients that may cause them being more vulnerable to doxorubicin. Because unfortunately, some patients are vulnerable and when they're given doxorubicin, they will develop cardiomyopathy, but there are some patients who do not develop any problems. So we hope that we can identify what causes the patients that are vulnerable to doxorubicin-induced cardiotoxicity, and find how we could possibly improve them being less vulnerable, so that we can continue to use doxorubicin, because it is a very good chemotherapeutic drug with very good results.
Any other questions? So if we don't have any other questions, I would like to thank Ludovic and Ms. Ghana. So Ludovic, that's wonderful work. I wish the best to see more. So it is.
Thank you.
It's my pleasure to welcome a wonderful friend of mine, Dr. Thorsten Kirsch. Actually, when Dr. Kirsch came to the States I graduated from Max Planck University, a really wonderful, excellent student in Max Planck. He published some really great journals, great papers in JBC, in top journals as a graduate student there. And then he joined the University of South Carolina. So when Dr. Thorsten Kirsch came to South Carolina, I was a graduate student, and we were in the same lab while he was a postdoc. So it's a really wonderful pleasure to welcome Dr. Kirsch. So following appointments at the University of Pennsylvania School of Dental Medical School and Pennsylvania State University College of Medicine, he was recruited to the University of Maryland. And later he became a tenured full professor in orthopedics. He did research at the University of Maryland Medical School. Dr. Kirsch joined the New York University at the Grossman School of Medicine in 2008 as vice chair for research in the Department of Orthopedic Surgery. And in 2019, Dr. Kirsch was appointed as professor of biomedical engineering at the NYU Tandon School of Engineering. So he currently acts as director of bioengineering and the liaison between NYU Grossman School of Medicine and NYU Tandon School of Engineering. So it is a really great pleasure. And thank you very much, my friend, for accepting to come and present at this seminar. Dr. Kirsch, it's all yours.
Thank you very much, Faxon. Yeah, I in fact know for quite a long time. He moved to Montreal and I moved otherwise I stayed in the States, and I'm currently at NYU Medical School, and you can see here, this is the main entrance to our medical school. Maybe, as you know, NYU went tuition free for medical students 2 years ago, and our ranking went now to the second best medical school in the US. So I don't know in Canada with tuition, but that's a way to get better and better and get the best students, I guess. So today what I want to do, I want to talk to about our peptide. It's a 15 mil RHAMM-Mimetic peptide, and we use this peptide in cartilage repair, and I will talk about the role of this peptide and what we know, what we think we know how this peptide might work. And this work actually was a collaboration between my group and Dr. Carmen's group. Dr. Carmen actually has was the previous chair of biomedical engineering and is unfortunately for me now retired, but she's still kind of active and comes once in a while to the lab to discuss ongoing projects. So just interrupt me if you have any questions, just feel free. Oops, I'm trying to move this forward. Oh, there we go. So these are my disclosures. We have a patent on this peptide, and the patent application is called Compositional Methods for Cartilage Defect Repair using the RHAMM-Mimetic peptide. So what I'm talking about today is cartilage repair, and cartilage repair falls under the category of regenerative medicine. So if you look up, what does regenerative medicine mean? So the definition is a process of creating living functional tissues to repair or replace tissue. I don't know, there comes this repair tissue or organ function lost to disease or injury. I know there's this poll here. I don't know how to get rid of this. Yeah. So if we look in orthopedic surgery, if we look at cartilage defects, this is still a major problem to repair cartilage defects. And you can see here, this is healthy articular cartilage, a smooth surface, and then you have these defects or lesions. It could be during aging that you get these cartilage lesions, or if you have injury to the joint. And unfortunately, even current surgical method, there's no good way to repair these cartilage lesions. And currently still the best method is to replace the knee joint or other joints with artificial joint, which is the joint replacement surgery. So if you look at the numbers here, in 2016, there were 700,000 knee replacement surgeries in the US. And the prediction is in 2030, there will be 3.48 million knee replacement surgeries per year in the US. And you can see how the huge increase, and if we continue like this, there won't be even enough orthopedic surgeons to do all these surgeries. So that means we have to find ways, biological ways, that we can hopefully repair these cartilage lesions. Oh, I cannot move my slides here. Oh, there we go.
Okay. Can you move?
Oh no, there we go.
Okay.
Okay. And also, of course, many knee joint injuries cause eventually the early development of osteoarthritis. And of course, there can be several injuries in the joints, especially in the knee joints. So you can rupture the ligaments, the tendons in the joint. You can have injuries to the meniscus, and you can have injuries to the articular cartilage directly. And all these injuries, even they're surgically fixed, in many patients lead to the onset of early osteoarthritis. So what are we doing currently to repair cartilage lesions or cartilage defects? So there are two main methods currently used by the surgeons. One is called microfracture, which is shown here. So microfracture is basically what the surgeon does. They clean up the defect site first, and then they drill these small holes into the subcondylar bone, as you can see here. So why do they drill these holes in the subcondylar bone? The reason is, since cartilage has very little stem cells, so the idea is that you release some of the stem cells, mesenchymal stem cells from the bone marrow, with the idea that these stem cells then migrate into the lesion site and repair the defect. Now there's another method which is called the articular chondrocyte implantation method. So unfortunately, this method is quite expensive and requires 2 surgeries. So in the first surgery, the surgeon takes a little bit of cartilage from a non-weight bearing area, then sends this piece of cartilage to a company. They isolate the chondrocytes, expand the chondrocytes, and then send the chondrocytes back to the surgeon. And in the second surgery, the surgeon takes these chondrocytes and puts these chondrocytes into the defect, and then closes the defect with the hope that these articular chondrocytes then repair the defect and form new hyaline cartilage. Unfortunately, there was actually a study which was published in 2016 and joined by a Swedish group, and they followed patients who underwent microfracture surgery and who underwent articular chondrocyte implantation for 15 years follow-up. So what they found that both methods were not very good in repairing defects. So you can see here, microfracture, the failure rate was 32.5%, and 48% of the patients who with non-failure developed osteoarthritis, and the articular chondrocyte implantation was even worse. 15-year follow-up failure rate was 42.5%, and 57% of the patients with non-failure developed osteoarthritis. So you can clearly see there's a need to develop new strategies to regenerate cartilage lesions after injuries or cartilage lesions in osteoarthritis. So what can we do here? So if we look at this, of course, everybody talks about stem cell. Should we use stem cells? But then the question is, which stem cell should we use? Stem cells from fat tissue, which would be the easiest way. But unfortunately, there are several studies have shown that these fat stem cells are not that optimal for cartilage repair. So the differentiation in the chondrocytes is not the best. Then from the bone marrow, the stem cells, people have shown they are better for getting into chondrocytes. So maybe that's the way to go, isolate stem cells from bone marrow and use them for repair. But then, of course, the other question is, do we need any growth factors to stimulate their differentiation? Do we need any scaffolds? What else do we need here? So lots of questions. So what we actually did, we asked a different question. We asked, can we utilize the endogenous stem cells which are in the joint? So previous studies have shown that the synovium contains stem cells. And of course, if we would do a microfracture, we can release some stem cell from the bone marrow. So the question is, can we utilize these stem cells and help these stem cells to repair the defect? In particular, we are interested in the paracellular matrix, which is the matrix directly around the cell. And as you might know, that the paracellular matrix plays an important role in cell homeostasis, cell differentiation, and cell function. In particular, we were interested in a abundant paracellular matrix molecule, which is hyaluronan or HA, and what role it plays in cartilage repair. So what is hyaluronan? Hyaluronan is a big glycosaminoglycan, and you can see down here, here's the structure of hyaluronan. So it's a oligosaccharide made out of this disaccharide as shown here. And the molecule can be very, very long. And so there could be hundreds and hundreds of these units put together to form hyaluronate. So it plays a very important role in cartilage because it can absorb a lot of water, and it also plays an important role in the paracellular matrix. Basically, it surrounds the cell and kind of protects the cell against cytokines and inflammatory molecules. So to show you that articular chondrocytes indeed have a hyaluronan in the paracellular matrix, we used the fluorescence staining here for hyaluronan, and you can see here the red is all hyaluronan around the chondrocyte. So you can see it really surrounds, it's in the paracellular matrix around chondrocytes. So hyaluronan, of course, can also bind directly to the cells through different receptors, and the main receptor is actually CD44. But there's another receptor for hyaluronan, which is called RHAMM. And this RHAMM receptor has been shown to lead to fibrotic skin repair, fibrotic or scar skin repair. And you can see this here in this cartoon. If you have an injury to the skin, what's happening, macrophages, M1 pro-inflammatory macrophages come in, and fibroblasts, they proliferate and secrete a lot of collagen. And it has been shown that these fibroblasts also then express RHAMM, the RHAMM receptor. And if you have this RHAMM receptor, there will be a fibrotic skin repair. And our colleagues collaborators in Canada, Eva Törlik's group, they have shown if you knock out RHAMM in mice, you get a tissue, a skin repair, which is less scar formation. So clearly showing that if you have RHAMM on your fibroblasts and other cell types, the RHAMM signaling through HA leads to a fibrotic or skin repair. So they proposed a model for skin repair in which initially you have inflammation with infiltration of macrophages, then which leads to the proliferation of fibroblasts, and then the healing. And again, if you don't downregulate RHAMM, you get this scar formation. If, however, you downregulate RHAMM in these fibroblasts and macrophages, what's happening, and you get a more scarless repair of the skin wound. So they approached us and said, look, what about cartilage repair? Since cartilage also heals like a fibro type cartilage, they said, why don't we try this peptide for cartilage repair? Maybe it's a similar mechanism like in the skin, where you get this fibrotic repair in the presence of RHAMM, but maybe in the absence of RHAMM, you might not get the fibrotic repair or fibrocartilage formation. And what they had a 15-mer peptide, which is RHAMM-Mimetic. So basically what this peptide does, it blocks the binding of HA to RHAMM. And they used this P15-1, which is called this peptide. They used it for skin repair and showed that also with using this peptide, there's less scar formation in skin repair. So based on this, we came up with the following hypothesis. So we hypothesized that in cartilage injury, what's happening first, you have an inflammation, which leads to the influx of M1 macrophages and RHAMM expression on chondrocytes and mesenchymal stem cells. And then RHAMM actually complexes together with CD44, the other HA receptor, and signaling of HA with this complex leads to inflammation and to the fibrocartilage formation. In addition, CD44 also is a co-receptor for TLR, toll-like receptor 2 and 4, which are known to cause inflammation. So the idea basically here is if we use P15, which blocks RHAMM, then HA just binds to CD44 and can cluster the CD44 as shown here. So there's no interaction of CD44 with RHAMM anymore, and also the interaction of CD44 with TLRs is inhibited. And then you can see here the clustered CD44 can bind HA, a nice paracellular matrix of HA, which then kind of protects the cells against inflammatory cytokines and other molecules like the DAMs. In addition, we proposed that P15 might help to polarize M1 macrophages into M2, the anti-inflammatory macrophages, the M2 macrophages in this environment would then lead to hyaline cartilage repair and not to a fibrocartilage repair. So this was our hypothesis and how to test our hypothesis. So we first used skeletal mature New Zealand white rabbits, as shown here. And what we did, we created a full thickness cartilage defect, a 4 millimeter full thickness cartilage defect. And we made 3 microfracture holes, again with the idea to release some stem cells into the defect site. Then we closed the injured joint, and directly after surgery, we did the first intrathecal injection of our peptide, P15-1, and we dissolved this peptide in high molecular HA. We used Orthovisc. Orthovisc is currently being used for the treatment of osteoarthritis, so it's being already used for the intraarticular injections in patients with osteoarthritis. So we mixed the two together and performed 3 intraarticular injections, one directly after surgery, a week after surgery, and 2 weeks after surgery. And our control rabbit just obtained high molecular weight HA without the peptide. And then after 3 months, we sacrificed the rabbits and looked at the repair in the knee joints. So that was our first set of rabbits we did. So you can see here, we isolated the knee joints, and that's the macroscopic evaluation of the repair. So you can see here the circle indicates the defect site. You can see here, if we just inject high molecular weight HA, the repair is not so good. So you can see some cartilage formation, but you still can really see the defect, while if we use the high molecular weight HA together with our peptide, you can see the formation of smooth articular-like cartilage formed. And when we did the histology, it was quite astonishing. You can see here, this is our control, just high molecular weight HA. So you can see a pretty poor cartilage repair, more or less fibrocartilage formed here. But you can see in the presence of our peptide, you see nice red-stained articular cartilage in the defect site. So this is the control, a sham operated. So you can see really nice hyaline cartilage has formed in the presence of peptide. We did also type 2 collagen and type 1 collagen staining. So type 2 collagen is the main collagen in articular cartilage. And type 1 collagen should not be in articular cartilage, it's only present in fibrocartilage. So you can see here, if we just inject high molecular weight HA, you see this fibrocartilage, you see type 1 collagen staining, and you see type 2 collagen staining. In the presence of our peptide, you see nicely only type 2 collagen staining in articular cartilage here in the repair cartilage, but no type 1 collagen staining in the repair cartilage. So that was quite promising to us. So now we started asking the question, how does this peptide work? So we went back into cell culture. So we isolated mesenchymal stem cells and took them, these stem cells in culture, and asked first the question, what happened to these stem cells if we put them in an inflammatory environment like we normally see after joint injury? So to mimic this inflammatory environment, we treated these stem cells with interleukin-1. And you can see here, if we culture these stem cells in the presence of interleukin-1, you see the upregulation of catabolic markers in these stem cells like COX-2 here and interleukin-6, and also the upregulation of matrix metalloproteinases, which degrade the matrix. MMP3 and MMP13 are usually upregulated in stem cells when they are cultured in the presence of interleukin-1. Interestingly, when we used interleukin-1 treatment and peptide treatment, you can see the catabolic markers were reduced, the mRNA levels and the MMPs were also hugely reduced in the presence of peptide. If we use peptide together with high molecular weight HA, you can see still the reduction here, but also here with interleukin-6 and with the MMPs. So the peptide alone reduced it, but peptide together with high molecular weight HA also reduced these catabolic and inflammatory markers in stem cells. What about RHAMM, the receptor for HA, and CD4, the receptor for the other main receptor for HA? So you can see here, if we look at the RHAMM expression levels, you can see interleukin-1 upregulated RHAMM expression in stem cells. And interestingly, the peptide reduced RHAMM expression in these interleukin-1 treated stem cells. Also, the main receptor CD44 was downregulated in interleukin-1 treated stem cells. So what I've shown you so far is basically that the peptide can reduce the catabolic events in stem cell if they're an inflammatory environment. And of course, we asked them also, can P15-1 directly stimulate chondrogenesis of MSCs? And indeed, actually, we found that it can. So what we did, we put these stem cells into micromass cultures and induced chondrogenic differentiation in the presence of BMP2 in the presence and absence of the peptide. And you can see here, this is Alcian blue staining. So the more blue you have, the more chondrogenesis. You can see the strongest Alcian blue staining you see in the presence of BMP2 and the peptide. It's clearly increased compared to just BMP2 alone. And of course, here this is the control, which has the least staining without BMP2 and without the peptide. We also analyzed chondrogenic markers, type 2 collagen, SOX9, and you can see both markers are upregulated, of course, with BMP2 and are further upregulated with BMP2 and the peptide P15-1 is shown here. And type 1 collagen, which is a marker for stem cells or fibrochondrocytes, you can see there's clearly a downregulation of the mRNA levels by BMP2 and even further by combination BMP2 and P15-1. So that shows us actually that the peptide is able to stimulate chondrogenesis in these stem cells. What about how does the peptide act on chondrocytes? Because if you want to repair the joint, you have to see also the chondrocytes surrounding the defect, how they behave, and normally they're also in inflammatory environment. That means these chondrocytes also release pro-inflammatory and catabolic factors. So what we did here, we used chondrocyte cultures, particular chondrocyte cultures, treated these chondrocytes with interleukin-1 to stimulate again the inflammatory environment, and saw what the peptide can do. Sorry, there's a misspelling. Before we call this NP, but that's the peptide P15-1 here. So what you can see here, the peptide alone was able to reduce the catabolic markers in these chondrocytes, which were treated with interleukin-1, COX-2, interleukin-6, INOS, MMP13, all reduced. And what was interesting is to see if we have the combination peptide together with high molecular weight HA that was even more powerful to reduce these catabolic markers in chondrocytes treated with interleukin-1. You can see further downregulation of COX-2, mRNA, interleukin-6, INOS, and MMP13 if we used P15 together with high molecular HA. What about the cartilage marker, anabolic markers? They were actually increased, especially in the presence of P15 together with high molecular weight HA. You can see an increase in aggregan synthesis and an increase in type 2 collagen messenger RNA. Okay, and then finally we asked the question here, what about conditioned media from chondrocytes which were treated with interleukin-1 in which were not treated with interleukin-1? It's actually interesting, and that's not shown here, but I tell you that conditioned media from healthy articular chondrocytes actually stimulate chondrogenesis of stem cell, while you can see here, if we use conditioned media from chondrocytes which were treated with interleukin-1, you can see actually they inhibit chondrogenesis. You can see the decrease in aggregan, SOX9, and type 2 collagen messenger RNA compared to the conditioned media from healthy articular chondrocytes. Interestingly, if we treated this interleukin-1 treated chondrocytes with P15-1 and used this conditioned media, you can see we could actually rescue the inhibition of chondrogenesis by this medium compared to the IL-1 conditioned medium alone. So that means actually P15-1 rescued the reduced chondrogenic differentiation of stem cells in the presence of conditioned media from IL-1 better treated human articular chondrocytes. So what I showed you so far is basically how stem cells, how P15 acts on stem cells in an inflammatory environment and chondrocytes in an inflammatory environment. But if we look at tissue repair or injury, we also have to look at the immune cells, and specifically the innate immune cells like the macrophages. So normally if you have an injury, the first reaction is inflammation and the influx or activation of pro-inflammatory M1 macrophages. And then ideally, you want to get to this stage where this M1 macrophages polarizes into the anti-inflammatory M2 macrophages, which then stimulate the healing process. So that would be the ideal. Unfortunately, often in the injury, this inflammatory phase stays way too long and then stimulates a cascade of inflammatory events which you cannot stop anymore. So again, ideally you want a short pro-inflammatory phase, which then converts into this M2 mediated anti-inflammatory phase and repair phase. So that's why we also got interested, how does P15-1 affect macrophage polarization during injury? So again, we first looked in culture. So we used the THP1 monocytic cell line, which has been well described to polarize into M1 macrophages and into M2 macrophages. So if you want to polarize them into M1 macrophages, you use these monocytes, treat them first with PMA to convert them into M0 macrophages, and then you use these M0 macrophages and add interferon gamma and LPS, and then they polarize into M1 macrophages. So that's exactly what we did following the literature. And we also cultured in the second step in the presence or absence of our peptide. And what you can see here, this is just showing you that it really, these M0 macrophages go into M1 macrophages as shown here. So we measured the mRNA levels of interleukin-6, and you can see here there's a huge increase in interleukin-6 mRNA levels after this protocol with interferon gamma and LPS stimulation for 24 hours. So interestingly, if we cultured during this 24 hours in the presence of interferon gamma and LPS with our peptide P15-1, what we observed that the M1 markers were decreased, as shown in the top figure here. So one is basically the expression levels of the M1 macrophages in the absence of P15, and you can see that all the M1 macrophage markers like CD80, CD86, and HLA-DR, which are all receptors on M1 macrophages, are decreased, while also the pro-inflammatory markers like interleukin-1, interleukin-6, interleukin-23, interferon gamma, INOS, MMP9, NF-κB signaling, STAT, the TLR receptors, and TNF-alpha, all these pro-inflammatory markers are decreased during this polarization in the presence of P15-1. Interestingly, M2 markers were upregulated in these macrophages in the presence of P15-1, like CD163, which is an M2 marker, and all these molecules. And also interestingly, here there's interleukin-1 receptor 2, which is actually decoy receptor and inhibits interleukin-1 signaling. So you can see in the presence of P15-1, there's clearly a decrease in M1 polarization markers and the pro-inflammatory markers, and the increase in M2 markers in these cells. So going back to our in vivo studies, so we did several rounds of rabbits with the injecting of high molecular weight HA and peptide, or just high molecular weight HA. Again, you can see here in A, that's a peptide together with high molecular weight HA, and this is just high molecular weight HA. Again, you can see the newly formed hyaline cartilage in the presence of peptide, and here there's this kind of fiber cartilage formed in the presence of high molecular weight HA. Again, here this is type 1 collagen staining. So you see no type 1 collagen staining in the repair cartilage, only in the subcondylar bone. Nice type 2 collagen staining in the repair cartilage. And here, in contrast, in this cartilage here, you see type 1 staining here in the articular cartilage, and you see type 2 collagen staining. So clearly showing us that this is kind of a fibrocartilage formed here, while this one is really hyaline cartilage. We also looked in the repair tissue in the subcondylar bone for markers for M1 macrophages and M2 macrophages. So for M2 macrophages, we used arginase 1 immunostaining. You can see it here in E and I. So you can see more positive ARC1 macrophages, which are M2 macrophages in the nicely healed cartilage with P15-1, compared to the cartilage which only got high molecular weight HA injections, so less positive cells for arginase 1 And then we use the marker staining for M1 macrophages, which is CCL10. So you can see less staining in the nicely healed one in the presence of P15-1, and much more positive cells here in the one which just received high molecular weight HA. And again, this is all the quantification of all the rabbits we did here. So again, the cartilage repair is much improved. Collagen 1 staining is improved, so that means it's much less collagen 1 staining in the articular cartilage in the P15-1 treated rabbits. There's more type 2 collagen staining in the P15-1 treated animals. There is less CCL10 M1 macrophages in the subchondral bone of the P15-1 treated animals, and more M2 marker arginase positive cells in the P15-1 treated animals. So that shows us clearly that the repair greatly improved. The inflammation was less, and also as shown by less M1 macrophages and more M2 macrophages, which I mentioned before. You need the M2 macrophages to help with the repair process. So to conclude, what I showed you is basically that under knee joint injury and cartilage defect, you have inflammatory conditions, which of course infect first the chondrocytes. These chondrocytes then release catabolic factors like COX-2, IL-6, INOS, and MMPs, which leads to cartilage degradation and eventually to post-traumatic OA. And of course, if you want to repair, the inflammatory environment also affects the stem cells. These stem cells then don't really undergo differentiation into articular chondrocytes. They more become like this fibrochondrocyte type cell, which lays down a fibrotissuous matrix, which is a mixture of type 1 and type 2 collagen. In addition, of course, these catabolic factors released by these chondrocytes further inhibit this stem cell repair process, and eventually you end up with this fibro cartilage, which of course can't fulfill its function. It will eventually fail, and most of the patients then develop post-traumatic osteoarthritis. If in the presence of P15, what we think is happening is P15-1 first inhibits the catabolic events in these chondrocytes, so less release of these catabolic factors by chondrocytes, so that means less cartilage degradation, and also it helps the stem cells to really differentiate into articular chondrocytes and repair the defect with new hyaline cartilage, which we hope will really then have the mechanical stability to function like hyaline cartilage, and hopefully then prevent the development of post-traumatic osteoarthritis in human patients. So that's where we are currently with our peptides, and I like to acknowledge the people who did all the work. So it was really a collaboration between my lab and my current members, which is Eugene Lee, she's a longtime technician with me, Emilia Bianchini, she's a PhD student, and two of our orthopedic residents. They all have to do one research year. So, and these Utash Anil and Charles Lin did the last set of rabbits, and previous research residents started the project in rabbits, and Natalie Dana and Brian Buttel, and a former postdoc of mine, Claire Short. And this is the Carmen lab, and especially Ashley Sin, who's here, did a lot of work on the macrophages. And then our collaborators at London Health Science Centre in Canada, Western Ontario, Eva Turley and Len Lut. And I like to acknowledge our funding sources. The project was started with startup funds from our CTSI, and then from NIAMS, and we had some funding from a startup company, Novare Pharmaceuticals. And thank you. And I like to take any questions you have. Thank you.
Thank you very much, Thorsten, for that really wonderful presentation and very clinically relevant, really wonderful, really appreciate. So any questions, you can either type in the chat or you can just unmute and ask. So Thorsten, does the peptide alone stimulate BMP2? Because you showed really nice work, which shows that it works quite very well together. But does it alone stimulate BMP2?
We don't know how it stimulates chondrogenesis. We don't know whether it stimulates BMP2 at this point. There are reports that HI1, especially high molecular weight HA, stimulates chondrogenesis, but it's not fully clear how. So we think if you have a stable paracellular matrix, it might somehow, we don't know how the signaling works, stimulate the chondrogenic process by itself. But again, we don't know whether it goes stimulate BMP2. It would be actually good to look at this, whether any of these growth factors are upregulated. Okay.
So I was surprised that you are the guru in type 10 collagen. But but you didn't check for type 10 collagen?
We actually we checked. There was in the micromasses there was actually less type 10 in the peptide.
Yeah.
There was some, but it was less than in the just BMP treated cells.
Okay.
And in our hyaline, in our tissue repair, we didn't find type 10. But also not in the high molecular weight HA group alone. So we never saw any type 10 staining.
Okay. Ms. Ghana, can you help with the reading in the chat? Ms. Ghana?
Hi, I think Ms. Ghana's having some internet issues, but I'm happy to take over.
Okay, yes. Yes, Ida.
So there's some questions in the chat. So thank you for a wonderful talk, very promising work. Do you know how soon after injury in humans it would need to be injected or applied? So do you envision having to re-inject or reapply multiple times until the healing is complete?
That's a good question. We also try to, or Mary always tell me, oh, we probably should try this, do just the defect without microfracture surgery and then directly inject, because she's thinking more about the military also. So basically, if you would have the peptide high molecular weight HA and the military, the soldiers, into the joint, you directly just inject this mixture. So we will probably try in the future. How many, we don't know at this point. We started with 3. We want to try maybe just 1, whether we can reduce it. So we don't really know how many injections we would need. Plus, we don't know how long it will be in the joint. The only thing I can tell you, we're working together with somebody from radiologist who wanted to use the peptide for imaging, especially imaging inflammation in the joint. And he showed that the peptide actually penetrates into cartilage and stays there for, I think they looked for 72 and 3 or 4 days. It stayed in cartilage for at least 4 days.
Oh, wow.
Yeah, very interesting. Thank you.
Any other questions? Yeah, 3-4 days, that's really impressive to stay in the joint.
No.
Yeah. Because that's a problem we've been finding with the company, that we need for our peptide, we really need to have some sort of scaffold and control release for the peptide. Yeah, the residence of the peptide in the joint. Yeah.
And look, also the peptide is pretty stable. So we one time let the mixture was nearly a half a year in the refrigerator. It was still
Okay.
the peptide. So it seems it's pretty stable peptide.
Okay. Have you tried to see its stability in serum, in blood?
No, our collaborator Len Lute tried, and he said you can't get this down, this peptide.
Okay.
It was stable for a long time in serum.
Okay. Okay. Nice. That's really nice. Any other questions?
I actually have a very kind of specific question about the part where you were discussing the conditioned media and how, you know, when the conditioned media came from healthy cells, it would influence it in, you know, in a positive way, and when they weren't healthy, it would kind of stunt their effect. So I was wondering if you could kind of comment on that, because I know that gathering, you know, exosomes and extracellular vesicles and anything that's in that conditioned media is it's very hard, and it's very hard to standardize. So I was wondering if you can kind of comment on that.
Yeah, I hope Foxen didn't mention me as a guru of matrix vesicles. Actually, when I went as a postdoc, we worked on matrix vesicles and mineralization. And actually, I'm going back on, you mentioned exosomes, actually, we're going back to the vesicles. So the only thing I know what we did, we used conditioned media and actually removed the vesicles. And the conditioned media without the vesicle was much, much less active.
Okay, interesting.
In both ways, in the good ways and in the bad ways. So what we think is that IL-1 treated chondrocyte release, what we say bad vesicles, and healthy chondrocyte release good vesicles. So I think the vesicles play a major role in affecting how other cells respond, because they are really the communicator. Okay, there are soluble factors, but we found that the vesicles were the most effective in this medium to stimulate chondrogenesis.
Okay, very interesting. Thank you. Thank you so much.
Yeah, you remember, Thorsten, how you know, all those work that you were doing in the matrix vesicles, and
Well,
people at the time didn't believe them.
that is
Now
correct.
it's all over the place. So yeah, so thank you very much, Thorsten, and for that really wonderful. We hope that you move it into the clinic.
Well, I hope so too.
Yeah, because it's quite ripe for the clinic. So thank you very much, everyone, and thank you for attending this seminar.
Okay, thank you.
Yeah, thank you, everyone, and thanks, Ida, and bye, everyone.
Bye.
And bye, Thorsten. Thanks.
Bye.
Bye.